Bindu, you are all. You want to unmute?
Okay, I think I'm muted now. Can you hear me?
Yeah, we can hear you and can see the presentation. Slowly but
Okay.
clearly. Yeah.
Good afternoon, everyone. My name is Bindu Poudel, and I am the extension plant pathologist and the plant diagnostician. I'm based in Yuma County. Today, I'll talk a little bit about Immunostrip. And I just want to put it out there that this is not sponsored or anywhere related to Agdia. Agdia uses I think they have some patent over the name Immunostrip, but here I'm using it more as a general term that the strips based on immunology. So just wanted to put it out there. Um, as I mentioned before, I run the plant pathology program in Yuma County. A big part of my program is applied research that includes applied work, field works, and lab works. We do a lot of field trials on fungicide efficacy. The part that extends and includes, of course, field visits, any questions or concerns that the growers have, and the another major component of the program is the plant disease diagnostic and disease diagnostic lab. We do both clinical as well as field diagnosis. As some of you may know, you can bring the sample. The lab is free. You can bring in samples, but a lot of times I have to do a follow up with field diagnosis, and I'll explain to you in a little bit how those two things are interdependent on each other. The lab, the Yuma Plant Health Clinic, is under the umbrella of Arizona Plant Diagnostic Network, and we're under the bigger umbrella of the Western Plant Diagnostic Network and the National Plant Diagnostic Network. Just to give a little bit about plant diagnostics, when you have problem in plants and you have to diagnose the problem, you split them into two major parts. It could be biotic that can be caused by fungi, bacteria, nematode, viruses, mycetes, insects, pests, and phytoplasmas. They are caused by some kind of biological organism. And there are some of the symptomology that you see in plant that is caused by abiotic measures like nutritional, weather damage, toxicity. So sometimes when I get samples from you, and then I have to do a follow up because these things, the biotic side, I might be able to isolate other than viruses. I might be able to, or nematodes. There will be the signs or symptoms of pathogen, and I can diagnose in the lab. But sometimes I will have sample that looks very much sick, but cannot isolate anything from it. And then that's when I have to go back to the field and see if it is anything other than biotic, like nutritional. I can look at the pattern of the symptoms in the field, the growth stage, and things like that. And most of the time, I'm able to figure out if it's nutritional or the damage or toxicity. So even though I am a plant pathologist by training, as a diagnostician, I deal with both biotic and abiotic cause. So for an example, the first sample, you do not see any signs or symptoms of pathogen. You see the symptoms in the plant, but you don't see the symptoms of pathogen. And in here, you can see the signs of pathogen, and all you have to do is look under the microscope, and you will see some biological causal agent in there. Just to give you a little bit more idea, this was some samples that I was working as a postdoc. I got these samples. It was a greenhouse, basil production greenhouse with almost 100,000, 200,000 plants, and they all had this symptom, which was uh, very much look like alfalfa mosaic virus. If you have worked or seen alfalfa mosaic virus in lettuce or in alfalfa, these are very striking symptoms, and it is very easy to pass saying it's alfalfa and try to forget about it. But sometimes the part of what we do, the research, other than just diagnosis, is sometimes you have to prove it to publish it, especially if it's some first time you're seeing it. So I was growing those symptomatic plants on the on those potting mix that you get in Walmart, which is supposed to have nutrients for 3 months. as they claim in the back. So I grow them, and then in less than a week, all the symptoms were gone. And that's when I realized it was not a disease, it was just nutritional disorder. So I had to go back to the grower and tell them that they don't have to discard their plant, and they just have to work on some nutrition, and they were able to recover their whole greenhouse of more than 100,000 plants. So when it comes to diagnosis, when you see a sick plant, me or you, anyone, uh, when you are trying to find the causal agent, you have to examine the host. You have to look for signs of pathogen, if you can see any. It is very important to know the application history of fertilizer, pesticide, herbicides, etc. Sometimes I have too many follow-up questions when you drop the samples. It is because sometimes that's how you know some of the symptoms that can be caused by excess or less fertilizer. If pesticides had been used, is there any residual effect of herbicides, etc. And then sometimes you have to go back to the field and observe the distribution of symptoms. Most of the times when you have uniform distribution, when all the plants are showing the exact same symptoms, chances of that problem being biological is very less, and the chances of being abiotic, like nutritional or wind damage or health damage, is more. But it is just not enough. That is why we have the lab, and you always have to confirm whatever you think it is by testing. So what do you need to do the diagnosis? A correct diagnosis is very important to manage the disease. As you can see in the disease triangle, you need to have a host that is susceptible, and then you need to have the pathogen that's virulent, and you have environmental condition that favors the host and pathogen to allow disease development. But if you cannot diagnose what the pathogen is, then you cannot do anything. Just the same way, you also need to know your host plant, and you also need to understand the environment. But correct diagnosis of the causal agent is the crucial part in managing a disease. So there are different kind of causal agents. Right here, we're talking about the biotic ones. We have fungi and oomycetes like downy mildew, powdery mildew, fusarium, which most of them in general prefer high humidity, their movement within the soil, they move within the field soil, and they're soil-borne. Bacteria, they like warm and humid environment, even though we do not have the humidity. We have some crops that are grown on sprinklers, like so we see a lot of problem of that in cilantro, spinach, where we are using sprinkler irrigation. We see some bacterial problems with pathogens because they are so small. You have to keep in consideration of the microclimate. So we may not have warm and humid, but we have greenhouses where it's warm and humid, and we have even plants with a um, with a heavier canopy. Underneath the heavier canopy, you can have warm and humid environment that favors bacterial development. And viruses, we see it fairly common. They usually rely on vectors most of the time, and you can also get viruses with the plant material movements. And nematodes, they are mostly confined in the soil and water movement. We thankfully we do not see a lot of problem in the desert Southwest, but we have had some few fields where we have had nematode problems. So how do you verify that the cause, the symptoms that you see is caused by a pathogen? So how do you know that? How do you confirm that you have a,
What are you trying to do? Is this
you see a symptom here, and then you think like, oh, that's alternaria and cilantro. Uh, as a diagnostician, I can tell that, but as a researcher to confirm it, so I have to be able to associate that every time I get this sample, I have to be able to associate this pathogen. I have to be, whenever I make a slide from this, I have to be able to see that. That's, I build up the association. And then I should be able to isolate it. Most of the fungi, they can be isolated. I should be able to isolate it and grow in artificial media. And then I should be able to inoculate it back to a healthy one and cause similar symptoms, and then be able to re-isolate. So this is how you prove that this is a causal agent, because you were able to induce the exact same symptoms by isolating the pathogen. So just to take you a brief, brief tour of what happens in the samples in the clinic. So we get a variety of samples, and it takes different length of time for us to confirm what the cause, what the problem was. This is onion, and it was pretty easy. It was a Stemphylium rot, and it just make a slide, and it's very easy diagnosis. This is cucumber mosaic virus, and it just takes about 5 minutes. About $10 in Immunostrip. This was something where we didn't see the signs or symptom of pathogen, so we had to do molecular diagnosis. We're still working on getting our lab set up for that, so we had to send it to another lab. I got about 1 week, cost about $100. This took a longer time, and it cost more than $1,000. We didn't have to pay. We have good collaborators, but that is what you would be paying if you have to pay. And we never got the result. We never got the exact diagnosis. And it was at the very beginning of me when I was in the plant, when I started the plant clinic. But over time, I have understood that this is actually a symptom of herbicide damage. No one knows for 100% sure, but this is what we're leaning towards. So that is the reason that sometimes we need to know the whole history. And now if I get a sample like this from you, and then if you tell me, what you have applied and what the application history, then I'll probably be able to tell you that we have had similar symptoms samples in the past. We tried and we did not get anything. So chances are it is just herbicide damage. So these are different kind of diagnoses we have in the samples. Moving on to Immunostrips, as showed about, it just costs like $10. It's rapid diagnosis. It's pretty much like pregnancy test, where we have 1 line, it's negative. You have 2 line, it's positive. So the concept is, as I said, very similar to pregnancy test, which is based on ELISA. Okay, I have the spelling wrong there, but it's enzyme-linked immunosorbent assay. So in the past, it's based on antigen antibody test. So the strips come with antigen that detects the antibody in the plant sample. In the past, these tests used to take a long time, anywhere from 48 hours to a week or even more. But because of those testing strips that we have in the market, they are very rapid. We have the test within a couple of minutes. So how to use it? Most of the testing strips, they come with a small buffer bag with those mesh bags inside, and they have buffer inside, which is to extract all the enzymes and the antibodies that you have in your sample. So it comes with the buffer bag and test strips. You need very little plant tissue. The rule of thumb is to have a plant tissue as big as your thumbnails. So what you do is you cut the top portion and then you grind it in the buffer bag. You can use any blunt material. I just use Sharpie because that's something I always have in my truck or in my lab. And then you grind the tissue, let it sit for a minute or so. And then after that, you insert the Immunostrip in this side and just let it sit. The solution will start moving up from here by capillary action. And then you get the results in a couple of minutes. So usually if you have 2 bands, it means positive. And if you have 1 band, it means negative. However, it's not always that simple that you get 1 band or 2 band. Sometimes you do not get any control line at all. So that means your test did not work. Most of the time, from my very own personal experience, it is because the strips were inserted too deep in the extract. So let me go back. So if you insert it here more than this, then it does not, somehow it interferes with that capillary mechanism. It doesn't work. So make sure you don't dip too much. Just a little bit at the bottom is enough. Sometimes the extract does not move into the strip or moves very slow. That means your plant material, could have used too much plant material. Sometimes plant have different phytochemicals that might interfere with the test. Sometimes viscous tissue, if you're using plants that are very high in wax or high in antioxidant, you might not get it to move as fast. Sometimes line is green or pigmented, also could be too much plant material. Or sometimes if you're using color plants like purple or orange colors, you may not have those pigments that you wanted. So it's always good to contact the company and see if they recommend using those colored plant tissue or not. Faded line, sometimes you can get very faded line. It could mean that your kit has expired, or sometimes it could just be the pathogen, the titer of pathogen is, the titer means like the concentration of pathogen is low in your samples. The pros, it's a rapid test. It's quick, it's easy. You get your results right there. Sometimes I take it in the field with me. It's just amazing in that aspect. It's pretty accurate. It is simple. There's no complicated process to do it. Pretty much anyone can do it. It is safe. None of the reagents that you use are hazardous in any ways. It doesn't mean that you can drink it or you have to drink it, but it is very safe for you to work with. There are a wide selection of pathogens that you can use them to. I will show you a list, and then you can also always go to their website and look for the list of tests that are available. They do have a good shelf life if you store them at 4 degrees. However, it is recommended that you bring them to room temperature before you test them. The cons, they might be expensive depending on how much your budget is. It ranges anywhere from $5 to even $15, depending on the amount of Immunostrips that you plan to buy. They are cheaper when you buy more. It works best if you refrigerate them while they're stored. So if you're living in a work truck at 120 degrees outside, they may not work as well, as good. They might give false negative if not used properly. If you like we do the troubleshooting, sometimes it might be negative, but your sample could still have the pathogen that you're looking for. It is very pathogen specific, which means that because it works on that antigen antibody concept, so it's very pathogen. You cannot really say that I'm gonna get this is for cucumber mosaic virus, and you should so for tobacco mosaic virus. It's not gonna work that way. It is very specific. And of course, it is not available for all the pathogen. There is, very limited. The number is high. I think AgDia has about 43 tests available, but there are thousands of pathogens that it is not available for. But whatever it is available for, it's one of the best tests. This is not a comprehensive list. It is mostly available for a lot of pathogens. A lot of you must have used in the in-person necrotic spot virus recently. It works pretty well for Phytophthora. I have used it for a lot of Phytophthora species. All you have to do is, it doesn't work on the soil samples, but you have to have some samples of the root samples, especially. I have done a lot of tests with that on citrus. The places where they have the test is, we buy a lot from AgDia, but there's also Eurofin also has quite a few tests available. They call theirs AgriStrip, and AgDia calls them Immunostrip. The last time I checked, AgDia has about 43 tests available. The Immunostrips you can buy themselves, or if you want to bring it to the plant clinic, we can test them for you. Just not the Immunostrip. If you have any diagnostic question for people with different expertise, all you have to do is go to Arizona Plant Diagnostic Network, and it will give you a list of labs that are specialized in pest pathogens. So if you have some idea about what your problem can be, you can contact those people directly, and you'll go to them, or you can contact any of us in here, and we'll always know if someone is better suited for that program for that problem than me. I will send you to the other diagnostician or other plant pathologist, and vice versa. So there's absolutely no conflict of interest between us, and we will make sure like you are directed towards the best resource. For submitting samples at the clinic, you please make sure like whenever you send samples, you use this form. Now we have the APHIS permit that we can get samples from out of state as well. This form is available in the Yuma Ag Center, Yuma County Cooperative Extension Office, online, the website, Arizona Plant Diagnostic Network. So just make sure you fill out all the information before you bring in the samples, and make sure you leave your contact information, especially phone number, because I would like to follow up on text if I have questions before I process or while I process your samples. All the updates about the program, all the IPM updates from Yuma. And these are the 2 emails that you can send email to subscribe yourself from the list. We send out newsletter every other Wednesday. And with that, this is all about the diagnostic and the Immunostrip for today. And if you have any questions, I'll be happy to answer them.
Thank you, Bindu. Again, if there is any questions for Bindu or for others, you can either unmute like now between presentations, or you can simply write them down in the chat box, and we can get them out if you want.